Okay, the title of my talk is The Enemy Within, revealing the presence of potentially zoonotic viruses from Philippine bats. You'll have to forgive me if my title is a bit ominous or medyo dark on title, but I think this has relevance to what is happening right now in the world. And of course, knowing that the SARS-CoV-2 has its closest relation to a bat that was captured in China from a rhinolophus bat, about 96% similarity with the bat coronavirus. Okay, right. For the overview of my presentation, so there will be five major topics that I will be talking about. First would be the bat diversity, ecology, and classification. Then I'll move into bats and viruses, and of course, the main talk would be the enemy within, viruses from Philippine bats. And then from there, I'll talk about our current efforts in bat virology research, and I will finish my talk with some research recommendations for various state universities and colleges to do in bat virology research. Okay, let's start. Right, so let's start with bat global diversity. So bats are the second largest mammalian order. So as of 2018, there are about 1,411 species known worldwide. And this represents about 20% of all the mammal fauna found on planet Earth. And it has a global distribution. It's found in all continents except Antarctica. And it can be found in lush forests and even into desert biomes. And it can appear in various shapes and sizes, starting with very small 2 grams up to 1.4 kilos. So some example of this would be your flying foxes, which are of course found in the Philippines. Okay, so bat ecology and classification. So before I will talk about the latest in classification. So ito yung usap ko muna is yung kung ano yung nakasanayan natin. Right, so bats in general are divided into two suborders, the Megachiroptera or the old world fruit bats, and the Microchiroptera, which are the microbats. Megachiroptera is represented by a single family, Theropodidae, whereas your microbats are represented by several families. So essentially all the families in the order Chiroptera or bats are, except for fruit bats, fall under the microbats. So for fruit bats, old world fruit bats, like I said, there's only one family. There's about 180 species represented by 42 genera. And with the name itself, they are fruit and nectar feeding. And they use mainly sight and smell to navigate and of course to locate fruits. But some genera, or at least there's one genera that uses limited echolocation. And with the name Old World, so it's only found in Asia and in the Indian subcontinent and in Africa. So for the microbats, there are 17 families, so more all of the families except for fruit bats. And then there are more than 1,200 species represented by 135 genera. So they have a varied diet. So you have insects. They also eat fruits, especially the New World fruit bats. And of course, some groups of microbats also drink blood. And they have this very sophisticated means of navigation called echolocation. So it's very, very, very developed in them. And they also have a worldwide distribution. But looking into the new classification, the new subordinal classification, what like I talked about in the last slide. So the bats are previously separated into just two suborders, Megachiroptera and Microchiroptera, with Megachiroptera not capable of laryngeal echolocation, which means that the one genus of fruit bat that is capable of echolocation does this by clicks with the tongue. And with the new classification, the fruit bat family, Theropodidae, has been lumped with Rhinolophidae, which includes Rhinolophidae or the horseshoe bats, the Posederids or the leaf-nosed bats, and the Megadermatids or the false vampire bats into one big suborder. This is called the Ynterochiroptera. And then the rest, okay, which is called the Yangochiroptera, and this includes all the rest of the families. And you can see here with the asterisk that these are the families that are found also in the Philippines. Right. So we now talk into viruses. So what you see here is the number of sequences that are virus sequences that are found in bats that have been found so far in bats. So far, the family with the most number of genetic sequences belongs to Coronaviridae. Of course, Coronaviridae. This is where the SARS-CoV-2, the SARS-CoV, and the MERS-CoV belongs to. And you also have Paramyxoviridae. Okay, and then of course you have Rhabdoviridae, which the rabies virus belongs to. So this normaties, if you add them all together, they're about like more than 12,000 individual distinct sequences found in bats. And if you see here in this table, the WHO actually presented a list, a priority list of diseases with pandemic potential. So you see here COVID-19, which is happening right now, the viral hemorrhagic fever, or the Ebola or the Marburg virus, the MERS and the SARS. The SARS happened back in 2002. It was the first pandemic of the 21st century, and then the MERS, what you call the Middle Eastern Respiratory Syndrome, it's in the Middle East. And then you also have the Hendra and the Nipah virus, and then the Rift Valley fever, the Lassa fever, and the Crimean Congo hemorrhagic fever. Right. And what you can see here with this, with the seven top priority diseases with pandemic potential, is that four of these viruses have bats as an animal source, or at least for COVID-19 hypothesized to be um, with bats as the original source. So this puts to the center stage the bats as a very important animal in conducting virology research. So this begs the question: how do bats live with so many viruses? Uh, there's global consensus among virologists that bats are the definitive reservoir of viruses. There's so many viruses known to them, like I showed you from the other slide. There are about more than 12,000 sequences found in bats. And there's a research, there's a paper published back in 2018 by virologists from China and Singapore, titled Dampened Sting Dependent Interferon Activation in Bats. So this was published in Cell Host and Microbe. It's a brief report. So what they found out that, well, you see high viral load could result in heightened DNA sensing, as you can see here in the illustration with your C gas. And this will lead to immune system overactivation. So, for example, in humans, so the production of interferons or IFNB, which is responsible for the immune protection for virus infection. So this is mediated by several proteins, STING, and then of course you have your S358. So there's overproduction, and then of course there's overkill of your immune system. So what's happening with COVID-19 actually is the immune system of humans are overreacting to this virus. But for bats, if you look in the pathway for bats, the sting dependent IFN or the interferon activation is dampened due to the loss of your S35A residue, or this is called the serine residue S358. So what happens is that there's no overactivation or formation of interferons. So what happens is that in bats, there's a balanced state of effective response, but not over response against viruses. So, and what we have to realize is that this has been going on in the bat's evolution for so many millions of years, and in more ways than one, the bats have learned how to live side by side with the virus inside them, which makes them the definitive virus reservoir. Okay. Okay, so with the main topic of my talk, so the enemy within. So what I will be presenting in several slides would be the results of our bat virus studies in the Philippines. This is essentially a consortium of research collaboration between UP Los Banos, represented by the College of Veterinary Medicine, the UPLB Museum of Natural History, and the Institute of Biological Sciences, and several universities in Japan. Foremost would be your University of Tokyo. So we started this research back in 2007, and we were doing that year in, year out up until last year. And so far, we visited 11 provinces and islands and resulted to 12 publications on bat virology research. We documented 8 virus families, 5 novel genotypes, and we tested on 12 bat species that tested positive for these viruses. Right. First off, what are the viruses that we detected? So first would be coronavirus. So this doesn't need to be introduced. So we know full well what's a coronavirus, or at least some of us, some of our fellow countrymen have been infected with coronavirus. So back in 2010, we published a paper entitled "Bat Coronaviruses and Experimental Infection in Bats: The Philippines." And we sampled bats in several localities in UP Los Banos and UP Diliman, and in the Biodiversity Management Bureau office. It's called the Ninoy Aquino Parks and Wildlife Center. So we sampled there, and we found 2 types of coronavirus, the alpha and the beta coronavirus. And for alpha coronavirus, we found that in UP Diliman. We isolated that from an Asian house bat, Scotophilus culi. And for beta coronavirus, we found them in several species of bats. All of them are fruit bats. And for example, Cynopterus brachiotis and Tinochirus jagori. So these 2 species of fruit bats are well associated with human habitats. They can be found in agricultural areas, even in urban areas. And as you can see as well in the table, there's also high prevalence, especially in UP Los Banos, for at least for Tinochirus jagori. So about 71% of the bats that we captured tested positive for beta coronavirus. So subsequent analyses of the genotypes that we did, the coronavirus genotypes that we found, revealed that the alpha coronavirus we identified here as Batco Philippines Diliman 1552G1, ang haba ng pangalan, no, 2008. It has a 95% similar identity with a Batcov from China, number A515 2005. For the beta coronavirus, we identified it as Batcov Philippines Diliman 1525 G2 2008. It has 83% similarity with the coronavirus from named Batcov HKU9. It's from Hong Kong. -1 China 2007. Right, and we did further work with coronavirus, particularly within Calabarzon. So we sampled in Timonan. So yeah, back in 2000, this was in 2012. So we published a paper entitled "Genomic and Serological Detection of Bat Coronavirus from Bats in the Philippines." So we sampled in 5 localities within Calabarzon, and we did RT-PCR, or genomic detection, and we also did serology testing using Western blot technique. As you can see in the map, we captured a total of 207 bats, over 200 bats, and for the genomic testing, we found that almost 30% of those bats that we captured tested positive for coronavirus, whereas 66% of these 207 bats at least had exposure to coronavirus or has neutralizing antibodies for coronavirus. Okay, the number 2 virus that we found, herpes virus. Right, so it's under the family Herpesviridae. Mammals are the natural host, and disease associated with this family, at least for humans, would be your Epstein-Barr virus and the Kaposi sarcoma. So the Epstein-Barr virus, also known as the human herpes virus 4, and when when it infects older children or adolescents, it's called infectious mononucleosis. So what happens here with Epstein-Barr virus is that there's active infection of your B cells, resulting to proliferation of the B cells, and of course, increased T cell activation, and of course with your antibodies. And this manifests itself with swollen lymph nodes, pharyngitis, splenomegaly, and fatigue. As for Kaposi sarcoma, what happens with this disease on humans is that tumors develop on skin and mucosal surfaces, as seen in the pictures. So you can see it in the skin, sometimes in the gums as well. Right. This was in 2009. So we published a paper and we detected a new bat gamma herpes virus in the Philippines. It was actually isolated from these diadems roundleaf bat, Hipposideros diadema, uh, and we named it as HDHV1, so which stands for Hipposideros diadema herpes virus 1. So we captured this in Malay, Aklan, back in 2009. And further phylogenetic analysis would be that the HDHV1 belongs to a group called Gammaherpesvirinae, in which the pathogenic disease to humans, the Epstein-Barr virus and the Kaposi sarcoma virus also are subsumed into. Right. The number 3 virus that we found, the Reston virus. Right, so this formerly known as the Reston Ebola virus. So it's actually with the name Ebola, so it's actually under the family Filoviridae, which where Ebola virus and the Marburg virus also belongs into. It was sorry. It was first discovered in a biomedical laboratory in Reston, Virginia, USA in 1990. It was actually from an isolated from a long-tailed macaque. It was imported from a monkey farm in the Philippines. So this was back in 1990, and the monkey farm was situated in Calamba, Laguna. So I think the farm was named for light, and I think it's closed now. And Reston virus is hazardous to monkeys. But for humans, it doesn't show any pathogenic manifestation. So wala talagang nangyayari sa tao. And back in 2008, there was one outbreak in pig farms, and which actually led to from the secretary, DOH secretary, to deliver a press conference that saying that people should not panic because the Reston virus is not pathogenic to humans. But on the other hand, well, the outbreak happened in Bulacan. But on the other hand, so what happened was there was a wholesale culling of pigs all across the farms in Bulacan. So as you can see here, so there was an abattoir or parang sinunog talaga lahat ng mga carcasses to leave out the traces of virus. But what happened as well is that some of the pig farmers, the pig workers, farm workers, were also infected with Reston virus. So 3 years after from the pig culling incident in 2008 in Bulacan, we published a paper, Reston Ebolavirus Antibodies in Bats. So this was the first time wherein this Reston virus was documented in a bat species in the Philippines. Um, Although we didn't provide any genomic evidence for the presence, so we provided neutralizing antibody evidences for bats. So antibodies were found in Rosettus amplexicaudatus. So it's called the Geoffroy's Rousette. This species of fruit bat is a cave obligate. So you can always find this. Well, you can always find the species most of the time in large cave systems all across the country. So in UP Diliman, we found one individual with Reston Ebola virus antibodies, and in Atimonan, Quezon, we captured about 6 individuals positive for neutralizing antibodies. So the fourth virus would be your hantavirus. belongs to family Bunyaviridae, which includes the Rift Valley fever, of course, one of the WHO priority diseases. And the harmful hantaviruses are from rodents, and this manifests either as hemorrhagic fever with renal syndrome or the HFRS, as you can see in the picture with extreme eye redness in the picture. And then you also have the hantavirus pulmonary syndrome or the HPS. You may probably remember last year back in March or even I think April or March when of course the news of the spreading of the coronavirus, and then of course and lots of people were have known already that the virus may have originated in China. So there was another virus actually that there was a mini outbreak that happened in China as well, and this was associated with hantavirus coming from rodents. But back in 2016, we already documented a genetically divergent hantavirus from a Geoffroy's Rousette. So again, it's Rosettus ole, Rosettus complexicaudatus. The same with the previous slides. So it's a novel hantavirus that we detected from this Geoffroy's Rousette, and we numbered it as QZNV MT1720. So we found this in Atimonan, Quezon. But on the other hand, or fortunately, it has a very low prevalence. After all our sampling on Luzon Island, in Mindanao, in Samal, and Talikod, and Panay Island, we only found it in one bat out of the 376 bats that we captured. And this our paper actually represents the first record of hantavirus in fruit bats. Okay, and then the fifth virus will be your orthoreovirus. Okay, so this was our paper published in 2017. So it's the first isolation and characterization of a theroping orthoreoviruses in fruit bats in the Philippines. So um, what what happened here? We found the PRV genome from two species of fruit bats, Rosettus again, and then Eonycteris spilea. So it's another nectar bat. So both of these species are found in caves, definitely in large caves, and we found this on in Davao region, from in Davao City, in Samal Island, and in the eastern side of of Mindanao. Okay, in Site A. Right. And then out of the 84 bats that we captured and tested, so 76 of those tested positive for PRV antibodies. And what we know so far from various literatures that PRV causes respiratory tract infection in humans. So the Miyazaki bee strain. So um, essentially, Theropin or orthoreoviruses, some of the members have been known to be zoonotic, and uh, and the manifestation would be, pag-ubo-ubo, sinisipon. So this can be reminiscent of what happened with SARS-CoV-2. So it all started with respiratory ailment and something like that. Okay, the fifth one, Henipa virus. Right. So you may have, I'm not sure if you may have watched this back in 2014. So this was 6 years ago in TV Patrol. So the report nila, and this was in Sultan Kudarat. So the DOH Region 12 reported acute encephalitis syndrome, which resulted to 10 fatalities there. So, and what happened there in Sultan Kudarat, was was chronicled, documented in a paper by Paula Ching of RITM. I think she's a nurse. I think she's in a different hospital now. So the title of the paper was "Outbreak of Nipah Virus Infection, Philippines 2014." So essentially, what happened was this was in Sultan Kudarat, Mindanao. So 17 people were infected that resulted to 9 deaths. This was due to horse meat handling and consumption. And some of the deaths were also from exposure to infected persons. So what happened here, yung nangyari, so yung mga nagkasakit from, kasi kung nagkatay sila ng kabayo and then they fed on horse meat. So inalagaan sila ng mga health workers, so they were brought to the hospital. And of course, yung mga nag-alaga sa kanila sa bahay got infected as well. And the 9 deaths that resulted were from acute encephalitis syndrome. And from the paper, it was hypothesized that the flying foxes or the big fruit-eating bats were the source of the Henipavirus. Of course, this was taken from previous studies done in Malaysia, in Australia, and Bangladesh, and in India. And during the same year, DOH, represented by many staff from RITM and I believe some field ecologists from UP Diliman, Dr. Aloy Duya, went to Sultan Kudarat to sample all the animals that were found within the area, within ground zero. So this was a report submitted to the acting secretary of DOH, and they found that using serology testing, they found that at least 6 of the large flying foxes, so large flying fox Pteropus vampirus, tested positive for Nipah virus. Okay, so fast forward in 2019, so we did a field work in Del Carmen, Siargao, back in June 2019. So we captured several bats there in caves, in agricultural settings, in coconut plantations, coconut areas. And out of the 9 species that we captured, so one species, Cypripsideros diadema, the diadem leaf-nosed bat, tested positive, at least 4 individuals of the 23 that we captured. 4 of those tested positive for HEV and NIV antibody test. And of course, we also did genomic verification, but they tested negative for the NIV. And then lastly, number 6, the last virus that we've documented so far. So this is not yet published. So we documented pox virus in Biak-na-Bato in Bulacan. So this was back in March 2020. So March 3 to be exact. So March 5 to be exact. Just a week before the national lockdown was in place. So we were quite lucky that kahit pa paano nakapag fieldwork pa kami before the lockdown was enacted. So essentially, we documented pox virus evidences from genetic, genetic meaning PCR, microscopy, electron microscope, and gross examination. So with the gross examination, and we found that on Cynopterus brachiotis wings, actually there were lesions and rashes present. And if we publish this soon enough within the year, so this will be the first record in Asia so far. There has been a record of pox virus in fruit bats. So this was, I believe, in Israel, at least in the Middle East. And we all know that pox virus belongs to pox viridae, which also includes the pathogenic smallpox and the monkeypox. So smallpox, of course, one of the most lethal viruses known to man, and has resulted to several pandemics back in the 19th century or 20th century, or even during the Middle Ages, that resulted to several 100,000 deaths. Right. What are our current efforts in bat virology research in the country? So, right, so we have now a project. It's just approved this last January. So we titled it "Development of a Simulation Model for Prediction of the Next Outbreak of Bat-Derived Coronavirus Infection in Humans." Haba ng title. And then perplexingly, we named the project, the acronym, the Robin Project. So So, well, the Japanese had other ideas on how to present an acronym of the project. I don't have any idea. Kahit i-palindrome mo yan, kahit i-anagram yan, hindi ko maintindihan kung bakit the Robin Project because of, I don't know, association with Batman and Robin. But what we're trying to do with this project is, of course, trying to derive a risk model. So of the areas that are, that can be potentially have, this outbreaks of coronavirus from bats. So this collaboration, again, we're continuing the collaboration between our Japanese researchers, a collaboration, of course, UPLB, Tokyo University of Agriculture and Technology, Okayama University, and University of Tokyo. So it's a 3-year project funded by the Japan Science and Technology Agency. So I think this is the counterpart of the DOST sa Japan. And in the proposal, we plan to do sampling of bats in multiple sites across the Philippines, and we already started work in Mount Makiling here in Los Banos, and of course within the surroundings of the UPLB campus as well. So we've done some we've captured bats inside a forest in Mount Makiling, near buildings in the campus, in some of the agricultural pastoral landscapes there. And we did some oral and rectal swabbing. And then, of course, afterwards, we released them. So as you see, these are the pictures of our recent fieldwork. And what I would have to say is that backfield or fieldwork for field ecology work, especially when dealing with wildlife animals, mukhang kailangan na natin ng extra precaution. As you can see here, what we we're donning PPE para kami mga astronauts, and of course we use gloves, we use nitrile gloves or surgical gloves as well. Tapos doble yung paglagay namin ng gloves. And what you have to realize is that last year we were not doing this at all. We were not. We were very, very, I don't know, parang medyo pabaya kami kasi kamay-kamay lang, yung huli namin, we don't have any mask on, we don't have any goggles. So, but now this year, well, we're taking the extra mile, doing the precautions as well. And IUCN, we also do this because in support of IUCN's recommendation that there is a probability that we can also transfer coronavirus to bats as well. So this is a reverse zoonosis or anthroponosis, as what happened in minks and in ferrets, and even in cats in other parts of the world. So there is a possibility, there's a danger, actual risk of tayo makapag-transfer ng coronavirus sa kanila. Okay, so we also have a project entitled Informing Biosurveillance, Contribution of Theropodid Fruit Bats to Viral Spillover in the Philippines. So this is a long-term project. It's going to go for 5 years. This is funded by the Defense Threat Reduction Agency of the U.S. Department of Defense. So this was just approved last January as well. So essentially, it's under the DITRA, Fundamental Research to Counter Weapons of Mass Destruction. So with the idea that the U.S. Department of Defense recognizes that there is a possibility that viruses coming from bats can be weaponized in the wholesale manner. So this is a consortium, this collaboration consortium between UPLB, RITM in Alabang, and National University of Singapore. So with our project leader, Ian Mendenhall, and my colleague and office mate and my boss, Dr. Grace Takuma, and Dr. Catalino Demetria, and Dr. Fidel Malbas of RITM, and of course, me. So this will be a very, very big in scope, very long, well, relatively long term, as we will be sampling bats monthly in 4 sites for 3.5 years. So what's going to happen here is that 36 plus 6, so we're going to be sampling for 42 months across 4 sites. So talagang dibdiba na pag-document ng mga bat viruses. and of course trying to pinpoint mga viral spillover events and their risk into crossing over to humans. Okay, so before I end my talk, so just a few recommendations on future possible research that can be done by especially for those who are watching today's webinars. Okay, so as you can see here on the map, these are the only areas that we have data on bat virus. When we started this back in 2007, there was only one paper on virus from bats. This was on Lisa virus or the rabies. So this was published in 2002. So and then afterwards wala na any publication until that we came on board and we did several surveys across the country. But nonetheless, even though we've been doing this for 14 years, 2007 to 2021, we've only surveyed so far 11 provinces and islands, as you can see in the markers. And we have a total of 79 bat species, and we only know of viruses that are found on 12 species. So 12 species pa lang yung na-document natin. So there are so many bat species out there that we don't have any data on. So especially as you can see here, maraming mga there are blanks or gaps, especially in Visayas. Virtually nothing is known in Panay or in Negros, in Cebu, Bohol, Samar, Leyte, in Palawan as well. We don't know anything about viruses from bats there. Northern Luzon as well. Sorry about the numbering. Number two, we also have to integrate in natural history studies with zoonotic research. So the value of vouchers. So kasama dito sa webinar na to si Dr. Gonzalez, si Dr. Jesmos. They are part of well-established museums. These people are museologists, so they know the value of vouchers. And of course, what I have to tell you about the value of vouchers is that there are so many data, that ecological data, reproductive ecology data, that are associated with a voucher specimen. And in previous studies, but not in our studies, in across the globe, nangyayari is some of the virologists capture bats, but pero hindi sila they're not keeping voucher specimens. So we're losing information about, let's say, through space and time. So what happened? Ano ba yung mga ecological information associated with that bat? Is the bat pregnant? What locality was it captured? Time of the year, or reproductive condition, or even the age class. So there are lots of data that can be produced with the synergy between bat virologists and natural history researchers. And for number three, it would be worthwhile, it would be very outstanding if institutions with virology research capability to partner with regional SUCs, state universities and colleges, and even private colleges with field ecology programs. So we know for the past several years there's the discipline on field ecology has been rapidly growing. So in several parts of the Philippines, so we now see several presentations from Northern Philippines, from Southern Mindanao, from Palawan, from Visayas. We now see several schools, several universities presenting papers on biodiversity. So what we see here is that talagang spread out, equally spread out, and in high numbers, yung mga nag-research on biodiversity. So for virologists or virology institutions across the Philippines, let's say, for example, the Philippine Genomic Center in UP Diliman, in UP Mindanao, Ateneo, UST, who has research capability, molecular research capability, to partner with them so that we can expand our search area and essentially expand our surveys of bat viruses. And some of the, of course, research areas of research that I would recommend would be, of course, first would be virus documentation. We really need to know what are the viruses from bats out there Kasi sa ngayon talaga, kulang na kulang talaga ang nangyayari. And of course, we can also work on synanthropic bats. Synanthropic meaning these bats are associated with houses or human habitations. So marami po tayong mga species na ganyan. Yung mga pinakita, like I showed you a while ago. So you have Sinopterus brachiotis, you have Tinochirus trigori, and of course you also have the Asian house bat, Scotophilus culi, which are found in attics of buildings and in houses. And we can also do research on tourist caves. So we can actually sample bats that are found in these tourist caves. Of course, these caves have been visited by the thousands. Like for example, let's say in St. Paul subterranean river or in the underground river. So the tourists that visit there are numbers to about by the thousands, and of course coming from different areas of the globe. So this can be a potential ground zero event because there could be a jumping over from bats because we know full well that bats are found in that cave. So tapos pagka nag-uwian na, so they will go to different places, and so the spread, the transmission, the risk of transmission and spread is there as well. And of course, we don't have any data so far for flying foxes, at least published data here in the Philippines. But we know full well that at least Henipavirus antibodies have been found in flying foxes in Sultan Kudarat. What I have to tell you is that Henipavirus or the Nipah virus has a very high fatality rate, ranging from 45% to 70%. So with the coronavirus that's happening right now, I believe the fatality rate is about around less than 5%. But unfortunately, of course, with the SARS-CoV-2, mataas yung R value niya, so madali talaga yung transmission. So we're now nearing about almost 1 million or more than 1 million deaths. Yeah. So with that, maraming salamat po. Thank you very much for inviting me to this webinar. So let us start working together. Maraming salamat po and have a pleasant good afternoon to all of you.